Recording. Again, welcome. It's the Walter Westman Lecture, which is sponsored by the Seattle University International House Alumni Association. It's an annual event in honor of Walter Westman, who was a resident at International House in the 1960s. He was on a Fulbright doing research in ecology. He died at an early age at 46, and after completing his master's at Macquarie University and a PhD from Cornell University in the United States, he is well known for his research and activism related to ecology and human rights. So we're delighted to have this lecture that honors him and provides a view into environmental issues important to the world these days. I'm going to turn it over shortly to our host, and then he will introduce our speaker. But I'd first like to do the acknowledgement of country. I live in Seattle, so I'm going to do it for me, but you can think about the area you live in. I live in Seattle on the historic and contemporary lands and shores of the Duwamish, Suquamish, Muckleshoot, and other coastal Salish nations. I acknowledge the forced displacement of Native communities from this land while honoring the endurance of the Duwamish people who still live here. If I can ask you to turn off, if you can, turn off your videos and audio so that we really get a good view of our speaker. At the end, you'll be able to come back on and ask questions and so on. With that introduction, I'd like to introduce Ian Gutierrez, who is a member of the SWIHA, the Sydney University International House Alumni Association, fondly known as SWIHA. He's a member of the committee who has been organizing the Walter Westman Lecture for the last couple of years and is already thinking about the one for next year. So we're delighted that he's taken on that responsibility. Let me turn it over to you, Ian.
Thank you, Dennis, and good day, everyone. I'm Ian Gutierrez, and as Dennis said, I am a member of the General Committee of the Sydney University International House Alumni Association. I lived in IH from 1996 to 97 when I was studying at the University of Sydney. It's my absolute pleasure to introduce someone today who embodies the very spirit of the Walter Westman Lecture on Science, Humanity, and the Environment. From a very young age, our speaker has done projects. By young age, like I think he was 6 or 7 years old when he started doing projects on marine science for the fisher folk in the Philippines. And his passion for the environment, his passion for marine science continues to this day. He works as part of the Global Wetlands Project based at Griffith University's Gold Coast campus. So for a Queenslander like me, I'm proud to have someone from Queensland present on Walter Westman Lecture for today. He has been involved in key projects that involve the application of automated technology in monitoring coastal wetlands for conservation. He completed a first-of-its-kind project assessing fish habitat use of intertidal oyster reefs at night using novel technology that combined non-invasive video surveys with bespoke infrared light rigs. And I think he will be explaining to us and even show us images of what they actually do. He's currently involved in the global monitoring of wetland biodiversity for conservation action, working with partners in 20 countries around the world. The project uses automated image analysis to understand animals that use wetlands and how they are affected by changes. It aims to highlight species diversity, monitor trends in wetland health, and provide advice for both the general public and policymakers to better protect these key ecosystems. His other project involvement include the automated monitoring of sea cucumbers and key fish species around Australia to guide the sustainable management of fisheries, as well as the monitoring of fish to assess the health of the Gold Coast's waterways. He holds a first-class honors degree in marine science from the Griffith University, where he also received the University Medal and the Head of School Award for Excellence in Marine Science. So it's my absolute honor and privilege to introduce today's Walter Westman Lecture for 2026, Mr. Diego Gloria. Diego, over to you.
Thank you, Ian, for the warm words. And I'm really honored to be here as well. Thank you for the opportunity. So I'm here to show everyone watching that automation is an up-and-coming topic that has tremendous potential. And today I'm here to show you how it's revolutionizing the way that we do, certainly the way I do marine science and conservation. So a quick outline of how that's going to go. A brief introduction, which Ian has probably done quite well, better than I could ever introduce myself. So that's great. And I'm going to walk you in through how we do automating marine monitoring and how that in turn supercharges research and conservation and what we do in our lab. So let's get started. As Ian mentioned, I'm a research officer at the Global Wetlands Project. We are also part of the Fish ID team. So those two labs are closely interlinked at Griffith University. And what that means is we use this technology to look at different applications, different marine applications. So at its core, I am a marine biologist. I am an ecologist. I like going out into the sea and answering all those questions. It's just that the way we answer it is then made easier by the use of the technologies I'm going to be showing you in this video. And that is all related to automation. And I graduated from Griffith University with a Bachelor of Science honors just last year and the University Medal. And on a personal side, I am passionate about the environment. I'm a keen fisher. That is how I got here. And here are some photos to show that. So really just sharing that knowledge with people through students, getting them into science is something I really like to do. And in my spare time, I go to fishing myself, go scuba diving, and really get myself in and on the water. And that's really how my journey to marine science began. I was really young. I really got into fishing. It was something that I always wanted to do with my family. And I said, okay, that's great. That's something I like to do in my personal time. But how does it affect other people? How can I turn this passion into something that's good, not just for myself, but can help our communities? And inevitably, when you do go fishing, you meet a lot of people. And in my case, I'm originally from the Philippines. I met fisherfolk, and I started to understand that their lives were so dependent on the water. And a lot of these artisanal people who are, fishing for their families, for livelihoods, small-scale fishermen, they're very respectful of our waters, and they know a lot more than I ever would about how to protect our waterways and how these ecosystems work, which made me realize that there's so much to learn from them. But also, if I pursue marine science, which is something I'm really passionate about already, it is something that could possibly benefit other communities. And I think that's a good enough reason to pursue anything. I did pursue it. I went to Griffith University. I ran a whole lot of projects as a student, and I liked university so much. I like that whole process of learning about our oceans that I decided to pursue an honors later on. And I did that on oyster reefs, which was really interesting. And I'm going to use oyster reefs as a case study of how we do, these ecological experiments and how our new methods can then expand to do this at a larger scale. When I started university, I had no idea what oyster reefs were, and I suspect most of the world still doesn't know what they are. And that's fair. The way I like to explain them is, if you think of a coral reef, an oyster reef is almost the same. The only difference that is, instead of the brightness, the hue, the vibrance, the clear water of the coral reef, with an oyster reef, it's quite muddy and stinky, and you can't really see much when the water's in there. But they're just as productive. And the truth is, we've only scratched the surface of how they work. But unfortunately, they've declined by 85% globally, especially since the start of the Industrial Revolution. And many are functionally extinct in estuaries, meaning they don't serve those functions of fish habitat and nurseries that we know that they do. So we're losing them without even fully beginning to understand how they work, especially in the tropics. So these photos here, I took the one on the left. That's in Sandstone Point here in Queensland. That's where I did some of my honors. And on the right is a photo of brim that I've taken from, I think it was Ozfish. So those are some fish that use these habitats. The good news is restoration efforts have been underway. So we're starting to understand how useful these reefs are, and now we're trying to bring them back. So there's a huge push, and the US has led this actually in the last 20 to 30 years, leading this whole restoration initiative. The other thing is, though, that effective habitat restoration requires the understanding and key integration of key animal users. So in ecology, we really had this whole field of dreams hypothesis for the longest time, which is essentially, you plant the trees or you plant the oyster reefs, and the animals will come. And that is true to many extents, but we don't really know perhaps the best way of doing this. What are the arrangements? How do animals actually use these habitats? I mean, if I was to use an example for humans, if we build cities, we don't just build buildings, we start to understand which buildings do we need to put up, and how close does a hospital need to be to the school, for example. And it's the same with any animal, and especially with fish. And the truth is, animals absolutely do use oyster reefs. Like I mentioned, they clean our waterways, but they also provide fishery services. So this is where fish are born, this is where they breed, this is where they live and hunt. All of those things occur on these oyster reefs. And to contextualize it, to put it into better perspective, like I said, I like to go fishing. I like to eat a lot of these fish. And so oyster reefs are used by many of these species that we consider to be economically important. So here in Australia, it could be like snapper, for example, whiting, brim, crab, squid, octopus. If you want to go down the more charismatic route, you have sharks as well. And the truth is, even if you have animals that don't use oyster reefs directly, they would probably benefit indirectly because the fish that they do eat comes from an oyster reef. And that's great. So I said, this is all good and well. What is a gap that we can solve? So in oyster reefs in particular, in many habitats in general in the marine world, we don't know what goes on at night. So I decided to study that. Like Ian introduced earlier, I made some bespoke camera rigs. So we found that during the daytime, we had absolutely high abundances of fish, high diversities using the reef, and at nighttime, it was completely different. There was a complete change of function. If you looked at an oyster reef at night and compared it to just a sandy patch next to it, there was no difference. There was barely any fish. Which sort of begs the question, what's going on? And darkness alters the value of habitat to these fish. And if you just look at daytime studies, which we have done for many years, it doesn't paint a full picture of ecology. And the analogy I like to use is, again, going back to humans, if you look at a place like, I don't know, Las Vegas in the US, or here in Australia, we have Surfers Paradise just down the road from me. The people you see there during the day, perhaps families and tourists, are very different to the people you'd see there at night, perhaps people just fresh out of high school or athletes celebrating a win. And it's important to understand those two groups of people if you really want to build a place that accommodates them. And that's the same for fish. You need to understand them to understand how the habitat works and how to protect them. So we need to monitor these habitats at night to understand how they function, how we can protect them. And the way we do this, at its very core, involves counting animals. And in this case, that's fish. And that helps us understand and manage our ecosystems. The United Nations Economic Program had a case study. They have many case studies, and this is one of them because they looked at marine protected areas and coastal patrols. And this is in a European area in Gokova Bay. And they wanted to quantify these benefits to make it tangible to a large group of people, so fishermen as well as policymakers. And if you look at their goals, nearly all of them involve quantifying, counting fish to some extent. So let's say increasing fishers, catches, and income. Yep, you need to count how much they're catching and how much they're selling that for. Biomass is counting the fish under the water as well as apex predators. Endangered species, seeing is believing. You need to have a tangible number that supports evidence that they're going up. And of course, the same is reducing invasive species impact. These are common goals across many monitoring programs and many research programs. So you need to be able to count fish. So how do you count fish? These are some of the habitats that I work in, that I've been in. So you can think of oyster reefs, that's in the top left, mangroves, rocky reefs on the right. And I'll just leave a few moments if anyone wants to think, how would you count fish in these habitats? So on the right, for example, those are gray nurse sharks, critically endangered species. Completely fine. I took this photo when I was with them. Of course, it does scare some people, and perhaps not everyone wants to get in the water with them. Mangroves, again, structurally complex habitat. It's really, really tricky to really get down there. And you can see the water, how muddy it is. It's not like you can snorkel down there and count the fish manually. So the truth is, I've had many answers to this question before. Some people suggested nets, and that's an idea. That's how we did it traditionally. Or you could send a snorkeler down, again, more tricky in difficult waters, or waters where you have sharks, let's say. Someone sarcastically suggested dynamite. I don't know how that came around. It was someone with a PhD as well, which was really funny, but of course he was saying it as a joke. And of course, we don't want to dynamite our ecosystems. But again, that has been done in the past where they've used different things. So the way we've traditionally counted marine life has evolved throughout the years. Initially, we had nets, like I mentioned, fishing, trapping. So with traps, you could just leave, for example, for crabs, you could just leave them out for a period of time, let the crabs fall into the trap, and then catch them. It's also a way that people catch fish. With nets, you could lay them across an estuary or a river like they've done here in this photo, and then hope that the fish swim by. You can count the fish that fall in, and then hopefully release them alive. Same with fishing. So especially for larger species like sharks and all of those bigger, perhaps more charismatic species, the way to catch them is you throw a line overboard with bait, wrestle them onto the boat, get all the information you need, count them, and release them. And if you're counting marine life, it can be quite invasive because you are catching them, you are stressing them, putting them out into the environment. What if they don't survive? And that is a real question. And even then, you're not really watching them in their natural habitat because you're interrupting that cycle of life. So over the years, we've said, okay, let's put cameras and sensors down as technology has advanced. And I'm just going to play these videos in the background. I'll mute that. But that is the camera that we put down on the Great Barrier Reef. And now you can see that these fish are starting to come to the camera, that we're not scaring them by swimming in the water with them. And we can start to count them. For habitats where it's a bit more turbid or we're not able to get cameras down there, we've also started to use sonar. And this was looking at walleye and freshwater habitats. And so again, that's non-invasive. You're just listening quite literally to what goes on underneath, and you can start to quantify fish. Great, so you've got this data. You've got cameras, you've got videos, you've got sonar data. How do you then interpret that? Traditionally, we used to count them manually. So that is a photo of me and a bunch of my colleagues counting that video you see on the left, in the middle of the screen. And you count the fish and you argue over hours and hours of what that fish is and how many there are and if you should have counted it or not. And then you put it into an Excel spreadsheet. And that is kind of where the point of, is it cost effective? Is it time effective? Comes into play. Is it consistent? Because you can see there are 5 people in that photo. And I can tell you right now that all of us had a different idea of what the species of a fish was. So that becomes a bit problematic. And it also begs the question, how can we monitor the environment at a meaningful and effective scale? Do you really have to hire 5 groups of 5, essentially, to watch videos at such a large scale? Because monitoring programs, they'll collect data at unprecedented rates. And for this one, it was a fairly small project. We only had 40 hours of video, but most studies, they'll have heaps upon heaps of them. So how can we do that meaningfully? And as we found, the answer is often automation. And the way that works, I think it's best to show you all an example, is we feed that video into a program that we train ourselves, and it can count the fish for us. Not just count them, but also identify them, even if there are different fish in the frame, and it works across different habitats. So you see these are mangroves, not a place you'd put a net in, and also muddier waters. And that is the beauty of automation, and in this case, fish identification as well. So it uses technologies like deep learning to extract that information. So how can it supercharge research and conservation? Like I mentioned, traditional methods are more prone to human error. There are cost implications as well. If you have to train people up or hire, let's say, an expert who's very expensive to watch 100 hours of footage, that can add up really quickly. Automated has its strength in consistency. If you feed 10 different videos to a program, it's going to identify a fish the same way in all 10 of them. It's less likely to have those human error. It's not going to fatigue because he's sleepy and he wants to have lunch. Now it's going to be consistent. And it can be quicker once your data set starts to emerge, because now that cameras are becoming more and more common, you and I could probably purchase a GoPro. To give a Sydney example, we had a colleague who said, Diego, I want to monitor fish in the Sydney Harbour because I want to show everyone that, the Sydney Harbour is well and truly alive. How would you do it? And I said, yeah, just get a GoPro off the shelf. And he gave it to a citizen scientist who was very passionate, dropped it. And then the nice thing about videos is it's a permanent record. Like these videos I've shown you so far, you can see them because they were taken on a camera. And if someone ever debates that the identity of a fish, we can look at that together. And then with the power of automation, that large set of data gets to be processed a bit quicker. So in terms of research, how do we actually do it? So there's this one study that our lab did, which had a question, which was basically, how quickly do we see a return on investment on mangrove restoration? Let's say I put $1 million into mangroves. How quickly is it before the fish that I want to eat start using them? And the answer is, they found it's pretty quick. The paper saw that the youngest forest that they restored was 2 years old, which is not that long at all. And so they found that it matched healthy mangrove forests. So these things that you have put money into all of a sudden are functioning like these healthy mangrove forests that have been there for, tens of years. And the reason we were able to do that is because we analyzed 280 hours of footage. And that's a lot of footage. This is actually just what was included in the final data set, but the test data and all of that, it was more like 300 to 500 hours, which is a lot. And when you think of that, when you have 280 hours of footage and you're doing it manually, you're not just watching it, you also have to pause, identify the fish, write that on a spreadsheet, and record that data. So conservatively, let's say you're taking twice that amount of time. So instead of 280, it's 560 hours of footage. That's equivalent to 4 months of full-time work, assuming you're not taking any lunch breaks. If you like Lord of the Rings, like me, you're watching the whole extended edition, the whole trilogy, 48 times over. And I like Lord of the Rings, but I'm not willing to do that 48 times for work. And so that's where automation really came into the fore, because those photos there are examples of the snippets from those videos. And you can start to train that model and then run everything you need through it. And that data will come to you thick and fast. Another good example is something we did in South Australia in Victoria, where there was this group that put shellfish reefs back in. And we said, okay, well, is it working? Are they attracting fish? And I'll show you a snippet of some of the videos from there. The answer was yes. They absolutely did attract fish. They increased productivity. And these are some of the commercially important ones, like flathead there. Those are baby snapper, squid. That's a goatfish. So we found that the reefs worked. And not only were we able to get the number of fish, we were also able to get the size of them in the way that we ran this study. Because the moment you start to get size, I like to say it's a force multiplier. Once you get the size of a fish, or indeed any animal, you can start to look at its life stage. Is it an adult that's there in its final stages of its life cycle and it's breeding? Or is it a juvenile? It was born in that reef, meaning it's an important nursery. That gives us more information on what animals are where and when we need to protect them, and the extent of our protection. And we can estimate the biomass of different species as well, literally the weight of animals that's using that area, and then use that to model the production of that value, which becomes more important when you're talking to policymakers. In this case, we found that in the reefs that were restored, if you estimate that, you can get up to 13,000 kilograms of fish produced per hectare every year. And that's not fish that have been attracted from adjacent habitats. No, that's the reef producing up to 13,000 kilograms of fish. So it's very important, and it shows that the restoration worked. And it makes that more tangible for policymakers because you've given them a golden number that they can use. So all good and well, you've gotten the fish, you've gotten the size and count. What else can you look from videos? What about behavior? So before I show this video, I want to say two things. Number one, it was great because we're looking at luderic, and these are oftentimes cryptic fish. They like to be a bit skittish when you snorkel, so it's really hard to observe them. And the behavior that you see here, I'll just play the video, is something that you don't normally see if you're in the water with them, and that's them feeding. So you can see there that the way they feed is they wrap their mouth around a blade of seagrass and then they just scrape it off all of a sudden. And that's something I've certainly never seen it while snorkeling with them. But because we put these cameras down and they're non-invasive, they can go about their daily life not worrying about a thing. And we can see that, oh, you know what? Luderick don't just live on these seagrass beds. They're actually feeding here. And we should probably protect them. So that is information that we can extract through automation. Like you saw just then that we have a confident level of identifying the fish and not mixing it with anything else. But we can also track the movement of that head and the speed at which it occurs and those patterns to extract fish behavior. Another thing that I'd really like to show, and I'll give you a chance to get involved here, really, is this is another kind of behavior that we were watching the video itself and we missed it with the naked eye, but an automated program flagged it for us. And I want you to see if you can get it. So we've got fish swimming, these are common probability, and all of a sudden they're gone. So what's going on there? So let's slow it right down for you. Right, you've got the fish, and then you've got a flathead. So the first time we were watching them, we were oblivious. We just saw fish, bang, no fish. And we missed it. And then we fed the whole thing, the whole video snippet into an automation program, the one that we trained. And because it looks at every single frame that our human naked eyes miss, it said, no. The reason that the fish left was because something else popped up. And that thing that popped up was this flathead on screen, something that is commercially important for fisheries and also recreationally as well. We target these. That it catches things that humans miss as well. Or if it doesn't necessarily catch them, it can at least flag them for us. And because, again, it's a video, we can rewatch it. And that is part of the beauty of it all. Moving beyond fish, some of our marine scientist colleagues are interested in crabs. And this is a cool program that my colleague Cesar Herrera has developed, and it's called Crab Spy. And you put cameras out there, and you literally just watch crabs go about their day, which becomes really interesting because instead of just getting counts, you're getting behavior. Again, you're looking at, okay, the crabs are here. We've got this amount of them, but how much of them are burrowing? How much of them are on the surface versus underground? How many of them are engaging in breeding behavior or mating, or they're fighting each other for this particular spot on the ground? And we can map that and again, feed that into a program. Because it's so, I wouldn't say easy, but it's easier to do this now, we can absolutely blow up the scale at which we do this because we're not spending our time watching the videos. Now we can spend our time getting into the field and putting the cameras out for the model to process. And that's exactly what we've done. This is a restoration site here in Queensland where there was a development and they're restoring this mangrove project. And we wanted to look at how the crabs are using it. So we can put all those cameras out, those icons there, feed them into a program, a little app, have a video of those crabs, and then get a list of species, the density, as well as the sizes of each species present in there. And we can make this so intuitive for anyone to look at, whether it's a scientist, whether it's a policymaker, or just someone who's interested in crabs. That takes it, that's all taken care of for us. So yes, automation works. Those are some few case studies. How do we actually apply it? So let's use that crab example. Again, we can look at the behavior, we can count them. What do we do with this? What we're doing is we're currently monitoring mangrove crabs all over the world. And I'll leave this video playing in the background so you can see why they're so bloody awesome. They're literally in the mud. They get stuck in, they sift through the sediment, they bioturbate, they introduce oxygen and carbon back into the soil. And they're a very good link between those processes and other animals. So they can be very good indicators of mangrove health. And from that, we can understand why a particular forest is unhealthy or what's causing its degradation. Just by looking at the abundances of them, looking at how they change over time. You can see here on the right, we've got different crab behavior. Some of them eat those leaves that they get on the surface. Some of them bring them back into their burrows. And both of those are very important when it comes to understanding how an ecosystem functions. And not only that, larger animals eat crabs as well. So you're also monitoring a key source in the environment.
Okay, Dave. Yeah, okay.
So on a global scale, that's what we do, is we monitor crabs, and we monitor wetland biodiversity for conservation action. So mangroves are perhaps one of the better understood coastal environments, and they are at the forefront of restoration and conservation projects. And like I mentioned earlier, less attention is placed on monitoring mangrove fauna. And there are quite a bit of mangrove fauna. You think of charismatic, economically important species like, sharks and turtles and dolphins, or many of the fish that you and I like to eat. Those have lived or will live at a mangrove at some point in their life cycle. And so if we monitor those fauna, or at least monitor the environment that they live in, we can start to get support for habitat protection and conservation. The other huge gap is that most mangrove information that we have currently is at the regional level, because let's say we've done a research project in one portion of Australia and we've blown that up to include the entire eastern coast. Or we've said that one project in Tanzania represents both the neighboring countries as well. And that's great, but it doesn't really mean that the data is locally relevant. What about that one estuary next to a school that the kid is interested about because he saw dolphins there before as a kid and now they're not there anymore? What's going on there? Your regional level data cannot answer that question. Or upstream, you've noticed that the river's gone brown and you're catching less fish because perhaps there was a sewage treatment plant there, but you have no evidence for that. So we need to do more things at the local level. So now we monitor that. All these crabs and other animals, we do it at a local level, but at a global scale. And that is something that's arguably impossible without automation, because now we've got data from all our partners coming in, and it's coming at unprecedented rates. Honestly, it's probably more data than I could probably work with myself. And currently we have 20 different countries involved, and we're scaling up. And the beauty of it is because now they don't have to be crab experts in the field. I know I'm not necessarily a crab expert. We can start to involve a greater diversity of people. So up here at the top, we ran a workshop in Durban in South Africa with a bunch of our partners from that region. And it essentially meant, we could give them cameras to go home. They could put that camera down following a very easy to follow set of protocols. And we could just say, send us the videos and we'll process them for you. And then from those videos, we've said, here's how the shift in community has worked. And this is what it means for your mangrove forests, whether it's healthy, whether it's improving or getting worse. A great example I have is at the bottom, we have a partner in the Philippines, and they are really, really great conservationists. They love the environment and they've been working in mangroves for a very long time. But they're not crab experts. They're actually bird experts, cockatoo experts. And because they already have that capability to monitor and they're in the field quite often, we could just tell them, what's something else you could tack on to conserve your forests is put those cameras out there and we'll process the data for you. And they've been so switched on because, again, you don't need to be a crab expert to get involved with this. So essentially what we do is we monitor animal indicators of wetland health. And this is akin to the proverbial canary in a coal mine. You can have a canary there that will tell you if it's safe to be in there. And the moment that canary dies, then, well, you're in trouble. And we do that to a greater extent now with many fish and many animals like crabs. And because we can do that at such a large scale, we can start to get a fingerprint of what a mangrove forest looks like and then compare them across the world. So again, we have what we call a global crab assay. And we call it an assay because everyone's doing the same thing across all locations around the world. And that means we can start to look at that data and analyze it pretty much in a very similar way. And that graph in the middle of the screen shows the heat signature of these natural sites. So if you've looked at sonar signatures, I don't know if you're like me and you like to look at those things of whales where it's like sound bites. It's very similar to that, except this is a heat map of the time they spent above the ground. And on the left, you've got the natural sites. It has a very unique and distinct fingerprint. On the right, you have that restoration site where it was under construction. So the crabs, you can see the heat map is a lot less pronounced. They're spending less time on the surface. And you can track these. You can track those little blips of when they're on the surface, what times of day, and how it's getting bigger and bigger and bigger, and start to overlay them and see how close that restoration site is getting closer to what we'd expect for a healthy mangrove. And that's all done through the power of videos and automation. All good and well. Those are crabs. Not everyone is interested in crabs. I know I'm more interested in fish, and so are a lot of other people. Some people could be interested, let's say, in Bengal tigers in the Indian Ocean. We have mangroves there. So we pair that with what we call a locally relevant animal of interest, because we don't just want to get scientific outcomes, we also want to communicate this to a larger audience. So if you tell me that the school kids in your region are interested in the number of flamingos, and we can use that to tell them how your mangrove next door has changed, we can monitor that for you. So some examples here in the Philippines, we're looking at rabbitfish, a very culturally and economically important species, because that's what the consultation of the universities has said. Fisher Focus, well, they've said, look, if you tell us mangrove health in terms of rabbitfish numbers and how that affects our income, that is a lot more tangible. And that's a win-win because, again, rabbitfish are good indicators of mangrove health. So that's what we monitor. Here in Australia, we do it with the estuary ray, which is slightly different. Previous studies have shown that the moment you see an estuary ray, it tells you that the catchment is healthy. It's associated with good sediment and good biodiversity. So we can use locally relevant indicators with consultation, which means that it's everyone gets a say in this. And that's probably one of my favorite parts about this project. Great, so that's conservation. What if, let's say, you were fisheries and you wanted to know if it was safe to continue fishing because you want to do it for another 20, 30 years? We do that with sea cucumber stock assessments. So this is also known as the bêche de mer industry. And sea cucumbers can be very important, economically, but they also churn out a lot of sand that you see on coral reefs. So there's both an economic and ecological reason to be monitoring them. And we do this with a couple of our partners, Fishwell and Macquarie. And you can start to see the indicator species. They've said these are the ones that they're most interested in for whatever reason, whether that's what they fish for or ecologically they're important. But they're fisheries. They don't necessarily want the ins and graphs and the back end of what's going on. They said, can you develop a little app, a little website dashboard? That means we can upload a snippet of our footage, and then you tell us how many sea cucumbers are in it. And it was a challenge, but we got there. We trained a model, and we made a dashboard that said, yep, you can just drop your file here that you've taken, or multiple files, upload them. The automated AI will look at them for you, and it will tell you what's in them to the species level and the counts as well. And they get those summary stats. They can put it into their polished documents and go to government and say, actually, even though we're fishing here, it turns out we've gotten more sea cucumbers, so we should be allowed to fish more. Or similarly, in the other direction, they've found that, maybe it's gone down, so maybe we should calm down a bit and let it recover so that we can continuously extract in the future. So that's another way of presenting results to a different audience. Other projects that we work on that are supercharged, I like to say, by automated monitoring, because this goes on at such a large scale, is we use fish species here on the Gold Coast as indicators of waterway health. So we've picked 6 different ones with the help of other universities that are indicative of, once you have these 6, then you know your waterways are in good shape. So that's great, because even if you're counting mangrove restoration and all that extent, again, it's amplified by the data of the fish actually using it. And you can do that through putting videos down. And again, we do it for other fisheries, assessing whether restoration projects work, and marine protected areas. So it allows for really adaptive management, because the data comes thick and fast, and we could put them out, send them back thick and fast, and you can adapt your policies accordingly. Another fun thing is that automation can extend beyond ecology. And this is a project that we call Beach AI. So here on the Gold Coast, a huge part of our industry is tourism. We've got, I think it's 33 beaches here. And so the city of Gold Coast said, you know what? We want to know what's going on our beaches. When are the people there? We need to allocate lifeguards. When is it going to be traffic? What weather conditions mean that we need to hire more lifeguards for this season? And you can start to extract that data. So these are drone flights that we do, where you can put them out there and understand not just how many people there are, but how many of them are surfing, swimming, resting on the beach, under their cool cabanas, fishing. And you can put this into a nice summary document, again, because your audience isn't necessarily scientists, it's policymakers. And we can say, okay, well, this is the number of people on the beach. This is the number of people in the water, which means you need lifeguards for them. This is how far they are. These are the towers that perhaps during winter, where there's no one there, you don't need to man that tower and allocate that lifeguard to somewhere else that's more popular. So that allows for adaptive management. It's also enhanced coverage from drones, and it's very, highly accurate. I think we found 10 million more people than with manual counts, something like that. And it's very cost effective. So I will mention at this point, when it comes to data privacy and security, those drones are flying so high that you cannot identify the face of a person. You can see what they're doing, but you can't identify them. I say that because one of our pilots was approached because the drone was flown up, and I think he was in the beach, and I think he was peeing or something. And he was very concerned that he got caught. And he said, no, are you going to report me? Can you see me? Like, no, we don't really mind what you're doing. We just see that you're there, but we don't know who you are. So if you hadn't approached us, we wouldn't have known. So it's also, very strict when it comes to privacy. And I think that's quite important. And I want to mention that. So when it comes to all people, yeah, you can look at weekends, when they're there, what season, whether it's peak season or, less so. Australia Day is a very popular time for people to go out, and we fly drones out then as well, so we can help inform policy. And that's really useful. And these graphs are great. But again, what if you're a policymaker who doesn't really want to see all of this? Well, dashboard. So it's another heat map that we can have of where the people are and how many there are of them and how close they are to the lifeguard tower, because sometimes that's just a bit more meaningful. And that's the visual that people need to make an estimate of what they need to do. And that's a good summary. And you can get these graphs if you want to, but otherwise you can get the heat map. So it's tailoring the results of that automation, that supercharged automation, into something that's readable for everyone. So I guess what I'm trying to say is results can be tailored to a wide audience, whether that's in the form of dashboards and you just want to upload your footage and get results nearly instantly, or you want to be a bit more technical and model the production of a fishery. Or perhaps you're a policymaker and you're a bit more interested in, yes, this is what's going on in terms of our crabs, but what does it mean? Does it mean that it's not healthy? Why is it not healthy? What do we need to do? Do we need to lobby for more protection? Or perhaps you're someone like me when I was younger who I wanted to know the trends, and I could really do with an app on my phone that tells me, yes, the number of dugongs has been increasing, so that's good. That means it's been working, or it's been decreasing, so I need to go to my local MP and talk to them about how much I care about this. So because we're using video and automation, you can extract this data and present it in a lot wider ways than you would with traditional counts. And again, they're a permanent record. So that dugong can be a photo of a dugong that was actually seen last week. And we have done that when we present our results. And that's really exciting because it makes conservation a bit more tangible and a bit more real for everyone. So automation presents exciting opportunities to achieve conservation and management outcomes. And this is my last slide, but I will finish by just leaving with some really interesting photos, some video snippets that weren't taken by me, but by other colleagues of what goes on here in Moreton Bay. And because we're catching this footage, we can start to quantify things and send them to the appropriate audience. And that's excellent. So thank you for listening, everyone. I'm keen to see if you have any questions. I'll do my best to answer them. And thank you for your time this Saturday.
Thank you so much, Diego, for a very informative talk. For someone who frequents the Gold Coast and spends time at the beach, now I understand why there are drones flying all over. And I feel safer now knowing that at least something is being done with the data. And now it explains many things too, and why they tend to change the number of people and where they go to specific times of the day or within, let's say, summer. So thank you for that.
Thank you.
I guess we have time for Q&A right now, and people have two options. You can either type in your questions if you're a bit shy or an introvert, or you can raise your hand and just ask your question. But I would encourage everyone to do so, because we rarely get this opportunity to speak to someone who's a practitioner in marine science who have actually applied AI and automation as well. For someone like me who's a bit on the older side of the demographic scale, it's interesting to see because the workforce is changing a lot. AI and automation has become more prevalent, and seeing it applied in this case is actually good. So feel free to ask questions, or if there is anyone who'd like to share their comments, feel free to do so as well. So the floor is now open. Don't be shy.
Well, I just wanted to say thank you very much. I found that absolutely fascinating.
Thanks,
and like
Ross.
and like Ian, very reassuring. And as a, I suppose ex-statistician, I am just fascinated by the power of these methods. It's terrific. So congratulations. I haven't got a question. Your presentation was so clear and so interesting. It was very fascinating.
Thanks, Ross, for the kind
And
words.
Ross, we know you're a well-known person in your field. That's a very big deal. So thank you for sharing that feedback.
It's excellent, really, because we have amazing statisticians in our team. I think that's our strength, because I'd like to consider myself more on the ecology side. We've got data technologists and scientists who work more on the technical side, and statisticians like yourself. And it's interesting because a lot of the time we're collecting so much data that we don't know what to do with it, because this is something that we never would have considered, and the literature is only starting to catch up. So it's really at the forefront. So really great to hear that statisticians like yourself are still interested.
Oh, thank you so much.
Okay, we have two comments in the chat box. I'll probably start out with a question from Doug. How do you validate the AI models? You mentioned the video counts were higher than manual counting. Perhaps the automated counting was incorrect. We'll start off with that. Yep, Diego.
Thanks, Ian, and thanks, Doug, for that question. Very good point. So we do this thing called supervised deep learning, which means we're not just feeding it willy-nilly into the model. We're making sure that we're ground truthing it. So a lot of the time, especially when I was starting out with our lab, my job was to look at a video, whether that's a fish or of the boats of the people, and do the traditional thing of, yes, that's this fish. That's 1, 2, 3, 4, 5. It's tedious. And that really led me to believe that, yes, I do not want to be doing this for a long time, but you need to still do it. And you ground truth that and you compare that to the AI's performance. If it's not good at all, then you can start to adjust it a bit. How sensitive is it when it's confidence intervals? Or maybe we need more data for this specific species because it's so rare. So with the beach, I think you're talking about with the video counts are higher than the manual counting. Yeah. So with the beach AI one, that's exactly what we did. So we went in and counted the number of people in a certain frame. We compared that to how the model performed. And then if it was pretty spot on, then we were happy with that. If not, then we'd go back and revisit. So the other thing that I didn't mention in the presentation as well is with the beach AI that we did, we also compared it to manual counting done by people on the ground, so pedestrian surveys. I remember that was my first ever casual job at the university, was standing at an imaginary line and watching people walk past and see how many of them had prams and how many had kids. And, that is tedious. And we found that maybe they were underestimating, and, no fault of their own, they're underestimating the amount of people on our beaches. So, you can compare it with your own expert knowledge as well as what's going on elsewhere in the field, and then come to a conclusion from that and then adjust accordingly. And the beautiful thing is, because it's a model, it runs on your computer. I wouldn't say it's easy, but it's more straightforward to go to it and just tweak a few, hypothetically dials on the knob and change a few lines of code to see how it changes. And we're learning more and more how to do that more effectively as well as we learn. So yeah, thanks, Doug.
Thank you, Diego. So there's another comment on the chat box. It says Mabuhay. So I suppose this is someone from the Philippines.
Yes.
Thanks for a terrific presentation. Are you getting much interest here and overseas? So I suppose here may mean both Australia or the Philippines and other countries.
Yes, thank you.
In the context of that question.
Yes, mabuhay. So with interest, we do tend to get quite a bit, especially when it comes to this large monitoring program that we're doing all over the world with mangroves. So that's how we have 20 different partners. It's really a collection of people who did approach us and are interested, and people that we've worked with as well. And we do have interest from like I mentioned, one of our partners is from the Philippines, and I'm so proud of them. They've been doing an excellent job, and I would consider them to be a bit of a success story, because it shows that they're bird scientists, but it shows that they can get their fingers stuck in with mangroves and crabs as well. So there's a lot of interest there. We had one from one of I think it was on the African continent. I can't remember which one off the top of my head. They had mangroves, and the nice thing about a lot of our projects is they're scalable. So we could give you a document that says how to do it, and it's usually in pretty basic language. And they said, oh, we can absolutely do that. We're the youth group and we just need funding to do it. We're going to apply for funding and we can get involved. I'm just reading the chat. So he says, my wife is from Pangasinan. Perhaps you can generate some interest there. Yeah, absolutely. So that's the beauty. So we can go, one of us goes around sometimes leading these conferences and we give a chat, whether directly or indirectly. And, a lot of the time we'll get approached afterwards saying, oh, can we do this project? Can we automate them? So yes, lots of interest and lots of opportunities. So excellent for conservation, I think. Thanks.
Okay, and we have someone who messaged me privately who has a question, but doesn't know how to raise his hand. So,
That's all right.
Steele, over to you. You had a question?
Hey Diego, it's Asil. So first, I want to congratulate you for such a brilliant presentation. So I've never realized that you're such a good storyteller, but it's also a testament on how brilliant you are and how passionate you are with the topic. So and basically, I feel more hopeful that someone as young as you can actually solve problems of the world. So my question for you is, what keeps you awake at night, and how would you plan to solve them? In terms of the environment, right? Not
Yes,
other
thanks.
things, but in terms of the
Thanks for the
environment, and now because you're solving problems through automation. So, but my question, I'm interested on what keeps you awake at night, and do you have any plans in solving them in the future? Thank you.
Yeah, thanks, Sil, for the kind words. I want to say first off that I don't think I'm solving the world's problems. Certainly not alone, and that's the whole beauty of being collaborative. And my colleagues give me a lot of confidence, which is why I love working with them. What keeps me awake at night? It depends. When I was doing my honors, a lot of what kept me awake was the logistics of it all. Because at the end of the day, we're still scientists, we're still field workers, we have to do a lot of planning. And a lot of the time it was just figuring out whether this field trip will go ahead. If it doesn't, if it gets rained in, how do we adapt? So there was that. I guess on the larger scale of how we're going to change the world, a couple of the things that have led me to jump up and write on a notebook, I think, were the nighttime things. Once I had an idea of what do we monitor in the nighttime, where should we put our cameras, there was that. I have a whole list on my phone of things I'd like to investigate. A lot of it has to do with how our urban waterways are changing in terms of the fish assemblages. Right now, we're also doing a lot of stuff with crabs and sediment, seeing how involved they are and how we can capture that on our cameras. And it's closely related to our global project as well. So it really does depend, I guess, on the time of day, whether it's, a lot of the time, I think it's nearly 2 p.m. for us here in Queensland, Australia. And I understand the whole difficulty of getting people from all over the world at the same time. And, on Wednesday, we had a meeting at 10 p.m. here because that's the time that worked best for our colleagues in England and Costa Rica and South Africa. That literally keeps me awake at night, but it's always so rewarding because you get to work with such a diverse group of audiences and communicate conservation with them. So that's some of the stuff that we do in our daily lives. I hope that makes sense and answers your question.
Thank you, Diego.
Thank you.
Thank you, Seal, for that question. Again, the floor is open. I know there are quite a few people who have dialed in from some universities overseas. I know there are people here from South Australia as well, Diego, who have dialed in.
Sure.
Yeah.
So this is Dennis, who you introduced Ian, and I have a question that relates to citizen science. How do you engage, or do you see engaging the public who have an interest but not really any expertise, but would like to be involved in whether it's counting things or giving you data? Is there a role that you see for those kinds of individuals?
Yes, thank you. And that's a very good question. And the answer is yes. So I'm not sure if I mentioned it. I might have mentioned it in passing earlier, but Sydney was a good example if they wanted to see how Sydney's waterways were doing. And these were concerned citizens who had been living there in many years. And one of our colleagues said, here's a camera, put it down so we can watch it for you. And they did. And because they put that camera down, and it's a citizen who, I think she was a champion kayaker or something, so she could go against the currents and deploy the cameras in a place that I would not have been able to kayak to. This is great because she gets to get involved and we get to use her muscle. So she got there, put the camera down, collected it after a week, and we helped her look through the footage. And they saw some amazing species, some more fish diversity, I think, than we expected. So that one showed that the Sydney Harbour was well and alive. And because of that video was there, I think she trimmed it and put it somewhere. She might have shared it with a group of students to really solidify the fact that, yes, this is videographic proof that we do have healthy fish here. So that's an example of citizen science. We also work with all over the world a range of practitioners who are citizens in their own right, not necessarily scientists, who are passionate about the environment, and they have more local knowledge than any of us. And I think that's perhaps been one of the gaps of science in previous years, is they're experts in one place and we send them to another, and science can come across as a bit more prescriptive and less engaging. So now, because we have these protocols and automation allows us to do things a bit more decentralized, we can be less prescriptive and really take their feedback on board. And we can say, look, you're the person who trudges into these mangroves every day to put out crab traps. What is it that you're interested in monitoring? And they can give us an answer and say, okay, this is how we might do that. And then we can tell them, oh, okay, it's a mangrove forest. There are snakes and stingrays and maybe sharks. What is the safest way to navigate this? I trust you to do it yourself. So they can do that. And we can really leverage that local knowledge and these local citizens. And once you start to do that, I think they get to be more involved in conservation, because now, they've got, what am I trying to say? Their skin is in the game, because now they're getting involved with conservation. So that's how we do it.
Great, thanks. And thank you for a wonderful presentation. Very interesting.
Thank you. Thank you, Diego. We have something from the chat box.
Okay.
I'll read it out. Greetings from the University of Batangas in the Philippines. Hats off to your comprehensive and meaningful presentation. We look forward to collaborating with you on research initiatives similar to the work you presented. Batangas offers numerous opportunities in marine science, especially since it's home to the globally significant Verde Island Passage, the center of marine biodiversity. Perhaps my simple question would be, how can learners be engaged in activities similar to what you do in the age of social media and AI?
Yes, thanks, Ethan. First off, I want to say Batangas, Verde Island Passage, beautiful place. I've dived there myself and I want to go back. So, and yes, Center of Marine Biodiversity. I've not seen that many fish in such a small unit area, and I do want to monitor it. So how can learners be engaged in activities? It really depends on, I guess, the age and the expertise level, and we can tailor it, right? So we've had partners internationally where a huge thrust of ours is, yes, we have expertise in automation and marine monitoring, but we want to be able to hand over that expertise so that you can continue to do it, because it has to be scalable. So a lot of the time that means workshopping things as simple as how to train an AI model, because again, you guys are the experts. In the center of marine biodiversity, there are lots of different fish and nudibranchs and lots of marine species that I could not keep on the top of my head. And there's no one better in that position than you to help us with that. So it would be a case of preparing an ID guide using your local knowledge and say, I could say that's a tuna. And you could say, no, that's not a tuna, because that is a very specific case to the Philippines and it's this kind of fish. And you can help with that. And then you can help us train our AI models. And it's literally, we use a bunch of programs to do that. And we workshop that as well. You said social media as well. The beauty of one of our initiatives is that global initiative, you've got a bunch of scientists in the room, and this has happened so many times, a bunch of scientists and nerds like myself who go in a room and talk about what we want to do and everything, the methods and statistics and all that. And then we have a science communicator. She has her master's. She's interested in science, but at a very larger level, and she's more interested in communicating it. And she will step in the room and say, I did not understand a single word of that. And if I'm your audience, you need to fix that. And she will help us do that with social media, with the way we promote, because I think it's so easy to get lost in the jargon and the technical side of things. And someone needs to really go in there and bring you back to earth and say, this is how you talk to people. So in social media, I think I'd like to think I'm pretty young, but I still can't navigate Facebook. I'm going to be honest with you. So that's why we get people who are more involved in that field to help us with that. So whether that's presenting our data that you've helped us collect and say the local schools, local government said, oh my God, the fish catch has declined over the years. We need to do something. That's one of the areas that we really appreciate that help.
got a question.
Yes.
Uh,
Sorry.
it's about Sydney Harbour. I
Yes.
was catching a ferry yesterday and I just saw a note on the wharf. If you live west of the harbour bridge, you must not eat the fish you catch and release. If you live east of the harbour bridge, you can catch the fish, you can eat them, but only to a maximum of 150 grams a month. How can our waterways be healthy if that's the case? And I know nothing about marine science. It was
Thanks. That's a good question, very locally relevant. I had a lecturer who actually mentioned that to me, and I could scarcely believe it myself. So I'm a marine scientist, but not in that space. So that's more of an ecotox question. But from what I understand, it has to do with the concentration of pollutants in the water. So as the further you go upstream, you have more agriculture, more sewage, more industry, and that starts to get diluted and filter out more as you head towards the ocean. So I think what they've done there, and again, not an expert in that field, but what they've done there is they've determined basically a very tangible structure. Said this is the bridge upstream or west of it. That's safe. It's not necessarily that bridge in particular, but that's the safe measure that the public gets. And that's where the dilution is it's safer if it's sort of more oceanwards from that bridge. So I wouldn't say there's different definitions of healthy, right? We can say healthy in terms of the health of the mangroves, which is the traditional sense of doing it, extent. We can also say healthy in terms of the fish. In my case, that's a lot to do with counting them. For an ecotoxicologist, that would be not just the counts, but also what's going on in their body and whether that's safe to consume. And they do this as well for whales. For example, if you have 40,000 whales compared to 1,000, that's great. But what if those 40,000 whales are mutated and incapable of reproducing? And that's what the ecotoxicologists do. So we work with them quite a bit to say, yes, these are the numbers. What is the reason for that increase or decrease? And then you send them out to do that. So another example is South Australia. You've got these algal blooms. We can tell you that the fish have decreased and where they've decreased because we have fine scale data. But then we need an ecotoxicologist to go there and run the lab tests. So it's very intersectional, this science, and it's great. So I'm sorry I wasn't able to answer your question exactly, but I hope that
No, that's very interesting. Yes, you have. Yes.
Thank you. We have another comment on the chat box, Diego. Dynamiting fisheries in the Philippines and Indonesia is a long-standing problem. Could you work on restoration and education? I guess maybe there are examples where you've seen in the work that you've done where restoration and education need to go hand in hand. And what's the extent of impact that both activities could do for the conservation and sustainability?
Yeah, thank you. So I think education is definitely the first step there. And I say first step like it's an easy one. It's not. If you think about dynamiting and a lot of destructive fishing practices, whether it's because it's the easiest way to extract fish for the most amount of profit, or perhaps certain groups, individuals are forced to that to provide an income for their family, there are many societal pressures on them. And education would be the first bet. So I'll give another example, which isn't necessarily dynamite fishing, but it's also destructive. And especially, in the Philippines and Indonesia, good examples of countries that used to extent still do catch sharks and rays and other endangered species. And the education bit for them was not telling them that what they were doing was wrong. There was a bit of conservation and telling them, okay, we need to protect these species. Why? But also pivoting that source of income from killing these animals towards, ecotourism is a viable alternative so that you're not out of a job when we tell you to stop catching these fish. That is part of how marine scientists work with policymakers and social scientists as well. Again, it's very intersectional. How we can help in the education side is really providing those videos that I showed earlier of, what these animals do and how they're important. We can really start to put an interaction of those habitats. If you dynamite them now and you catch maybe 10 fish. Next year, you'll catch 1 or 2 or none. That can be made more tangible through the power of video and storytelling and science communication. And then from that, once we stop that stressor, we can work on what you mentioned is restoration, and we can measure the impact of that. So like the paper I showed you earlier, after 2 years, have the fish returned. If they haven't, then well, we know they can. So what's going on? Is someone still dynamiting illegally? Or is it because we protected one stand of mangroves, but everywhere around it, people fish out? So the fish have no pathway, I guess, from where they're born to where they live in the future. And that is that whole issue of what we call habitat connectivity is still a gap in many areas. So we can definitely do the research, and that can power the education as well through the videos we have and the communication. So there is a way to do it with local practitioners.
Diego, I have a question on the use of AI and automation, because you did say that you teach the machine or the program on what kind of fish, how many fish, you account for the statistical deviation or the confidence interval that you're aiming for.
Yes.
After how many hours of videos, or I don't know what data inputs do you have, would you say you have higher confidence? Because you did say that if, let's say, a local community would like to monitor the health of the mangrove by introducing or looking at a new species, for example, you can't conclusively reach a comfortable statement of with much more confidence if you don't, if statistically it won't make sense. How would you build confidence through time? And at what point can you say, yeah, we can now comfortably say and conclude that this is what happens, this is the pattern that we have to watch out for?
Yes, thanks. That one really does come with experience, and the more projects you do, the more likely you're able to build a workflow, as with anything. So how many hours of footage? It depends on the animal. So the one we like to say is here in Australia, we have a very common species called brim. Everywhere, all in your waterways, you're likely to see it in or tilapia, maybe in some countries, you're going to see it everywhere. You can easily train a model on that, and it will probably nail it within the first few hours because you have so much footage of it. And then the moment that we're asked to look at a more cryptic species, let's say flathead, because you don't see much of them in the cameras to begin with, you need to start to really spend more effort in terms of looking for that footage and extracting every single frame. So let's say with brim, you could look in one every 5 seconds. That's fine because it's common. Or one every 10 seconds. With more cryptic species, you're really looking into the milliseconds. If you're capturing videos, what's the saying, like 30 frames per second, you're looking at every single frame because you're making the most out of all that data. And that changes. So the model that we would use for a fish on the Great Barrier Reef is very different from the model that we would use in a very muddy estuary here on the Gold Coast. So it does depend. And when it comes to those endangered species, I think with a lot of animals, we have indicator species, like those crabs I mentioned, like those rabbitfish or flamingo, and those are easy to see. And we call them model species, model animals, because that's what we know is an indicator of everything else. But the truth is, with the amount of animals in the world, it's so hard to distinguish between a few of them, because some of them literally differ by the number of teeth that they have. And you can't see that on a camera, admittedly. So with more visual cues, it's easier. Like if you guys know what leafy sea dragons are, they're kind of related to seahorses. Some of them differ in the number of spots that they have. So you can use that to not just count them, but also get the specific individual. Same with whales, where the fluke, humpback whales have the back fluke. That is like a fingerprint. That's unique to every whale, and they use automated programs to deal with that. So it depends on the animal. With more cryptic ones, sometimes you just have to go into the field and collect them. And I was actually, before this talk, I was reading a paper that a summary that they've found 10 different species since the start of the year around the world. And that one, you really have to go out and collect data. So nothing will ever replace expeditions, I think, but monitoring at such a large scale to keep our waterways healthy is something that we're getting better and better at.
Thank you, Diego, for answering that.
Thank you.
Unless there are any further questions, I would like to take this opportunity to thank you for your time today, Diego. It's been very informative. For someone like me who's worked in banking most of my life, I learned a lot today, and it gives me comfort that the future generation is in good hands with people like you, applying both technology and science to conserve the future for humanity and for the future generations. So someone says here, thank you, Diego, from Sydney. And I know people from South Australia, the Philippines, all over the world who have joined us today. So in behalf of the Sydney University International House Alumni Association, thank you for being the 2026 Walter Westman Lecturer.
Thank you,
Dennis,
everyone.
would
Thanks
you
for
like to
having
say a
me.
few words before we end? Dennis?
Yeah, sorry. I had to find my mute button. I just want to thank Diego for being available for this. Thank you, Ian, for organizing it. Thank you, everybody, for being here. I originally didn't introduce myself. I'm Dennis Schatz. I'm the president of the Sydney University International House Alumni Association. I'm delighted to be able to continue these programs while we're redeveloping the International House at the University of Sydney. For those people that get our Alumni Association newsletter, I hope to have much more information in the future about the redevelopment of International House. Again, thank you everybody for being here.
Thank you. People can go, but Diego, there's one question that one person is asking.
Sure.
Dennis, you can stop the recording if you'd like to. I think there's a practical question.